The broadcast is now starting. All attendees are in listen-only mode.
Okay, hello everybody. Welcome to our European Industry MedDRA User Group webinar today. I'm your Chair, Chris Scholes from the EU MedDRA User Group, and we have Destiny Hutchison, and Brian O'Hare from the MSSA providing us technical support. So a quick housekeeping slide before we get started. So for anyone who's new to this webinar program, the instructions on how to make questions are here. So you can see how to submit questions. So pop out your control panel, which will be in the upper right corner of your screen. Enter your question into the question box. And then obviously when you've finished, hit send. And what we will do is when we come to the Q&A part of the session, we will start to run through the questions. Hopefully we will be able to answer questions, but if we don't, we will endeavour to get back to you after the presentation. Okay, so a couple of frequently asked questions. So will you be able to get a copy of the slides? Yes, they will be available. I believe some of the slides that will be presented today are already available as downloads. Additionally, is this a recorded webinar so you can pass it on at a later time? And the answer to that as well is yes. And you can locate that with the link at the bottom of these slides. So before we get started, a quick welcome from our industry user group. So we can see our current members there. For those of you who are returning attendees, I'm sure a lot of these faces are familiar. For those of you who is your first webinar, then welcome to the group and a big hello from all of us. So before we get started, a couple of polling questions we just wanted to run through. So this is our first question. So I believe the poll should now be open for this particular question. We have around about 30 seconds to do that, and then we will move on to the next one. Okay, so I believe that one is complete. So moving on to our next polling question. Here we go. As we go through these questions, I'd encourage everyone to reply because these help us sort of, judge our audiences and enable us to determine what kinds of presentations we can provide in future sessions. Okay. So I think we should be down to our third question. So yes, please have a go at this one. Okay, so I believe that is it for the polling questions. So we shall now move on to the part that we're all here for. So the agenda. So today we have presentations from several people from Bayer and Caroline from the MSSA. So the topic of the presentations today is enhancing the quality assurance process in medical coding. So the results of an experiment on automated visualizations to detect and identify coding inconsistencies, which I personally am very interested in listening to. So here's our presenter today. So starting from the left, we have Marit Weiss from Bayer. So she's a medical coding product lead within the R&D standards expert group. She's responsible for medical coding strategy and pharmacovigilance in clinical trials, including tools and process maintenance, development and improvement. We also have Adrian Robel, who will be presenting a live demonstration after the slides. And then we have Uta Getachew, who is R&D standards expert in medical coding, responsible for the medical correctness and compliance with the regulatory requirements in MedDRA maintenance and upversioning processes, documents and applications at Bayer used for MedDRA coding in pharmacovigilance and clinical trials. And obviously, I believe most people would recognize Caroline Wilson from the MSSA. I believe over to the presenters.
Caroline, we can't hear you.
Yes, I was going to say, Caroline, you may be on mute at the moment. We can hear you, Caroline. Welcome.
Oh yeah, now I was muted by the organizer. Sorry for that.
Not a problem. It wouldn't be a meeting without some kind of muting issue, so that's not a problem at all.
So again, I start from the beginning. So thank you, Chris, and hello to everyone. So I would like to give you some details about the data set that Marit has used for her visualization experiment. I'm a member of the ISOP medication errors special interest group, and together with 3 other colleagues, we decided to look into the quality of coding for vaccine medication errors during the COVID pandemic. You see the contributors here. Katharina and Joan are also members of the ISOP group. Here they are. And Nicolas was working at the Sanofi Global Pharmacovigilance Department like Katharina and supported our activities. And the objectives, methods, and results of our activities were published in the Drug Safety Journal in 2023. Our objective was to review a sample of verbatims describing medication errors that were reported for the COVID vaccines available at that time point. Only verbatims that were coded to MedDRA terms in the high-level grouping term medication errors and other product use errors in the MedDRA hierarchy were considered. Our data set included 1,500 verbatims with their MedDRA codes, all anonymized without any demographic information or access to narratives, and without specifying the relevant COVID vaccine. The coding quality of the sample was assessed by 2 MedDRA coding experts applying the 4 eyes principle. The data were drawn from Vigibase and included data reported up to the 25th of August 2021. And I would really like to sincerely thank the colleague at the Uppsala Monitoring Center, UMC, for providing us with the data. Just for your information, if you are not too familiar with what the UMC does and what Vigibase is. The UMC is maintaining and managing Vigibase. the global database of the WHO. It contains reported potential side effects of medicinal products that are shared by the national PV centers of the countries that are participating in the WHO program for international drug monitoring. The UMC itself is not doing any data entry or manual coding for the reported cases. Data entry and coding is done by the competent authorities which are reporting the case, meaning that the MedDRA term selection is in the hands of the national PV centers and not in responsibility of the UMC. Important to know. And here are the final results of our review. Of the 1,500 records, approximately 1/3 here was not accessible because the verbatim was missing or could not be interpreted. Of the 2/3 that were finally assessed, about half were accurately coded. Here they are. The other half was not. In most instances, the wrong PT has been assigned. And the most common PT or error was the assignment of two vague MedDRA terms like PT medication error, vaccination error, or also product use issue, for instance. And of course, it was an error because the verbatim provided more specific information. And despite this information, the verbatims were coded to these vague terms. When we discussed these findings within the ISOP group, a colleague from the FDA, who is also a member of the group, confirmed that the FDA makes the same observations in their PV database. So they also see a lot of vague terms that are assigned to verbatims that are describing the related medication error or vaccination error in much more detail. So in essence, we were able to show that half of the records that could be assessed were correctly coded, and that the incorrect code assignment in the other half was in most cases due to the selection of MedDRA terms that were too vague. Poor quality of data extraction, you all know, and too vague and incorrect coding, of course, hinders reliable and reproducible safety data analysis, not only for medication errors. This was especially critical in the time period of our assessment because the safety profile of the COVID vaccines was not yet fully established. That is why we decided to publish our findings to raise the awareness for the importance of good quality case documentation and coding. And because it is a crucial prerequisite for the assessment of cases, and in case of our data, for the safe use of COVID vaccines. So here are the final conclusions, pointing at the relevance of the quality of medication error documentation, data extraction, and coding. And we also provided references to MedDRA training opportunities and the ICH endorsed guidances for MedDRA users. So this was just a short introduction so that you know what kind of data MARIT has used. And with this, thank you for your attention, and I hand it back to Chris.
Thanks, Caroline. So I believe our next presentation now will be over to MARIT.
Thank you. I hope my screen is visible.
It is, and we can hear you, so
okay.
Good to go.
Great. So also a warm welcome from my end, and thanks a lot for your interest on this webinar topic. As we, the medical coding team at Bayer, we would like to present today the results of an internal experiment where we have used the data described and explained and introduced by Carol Ann, because we want to see, is there any possibility, using visualizations to help in detect or identifying coding inconsistencies. So we can go to the next slide. Let me check. Here, should work. As mentioned, right, so this is now the main part, and then after this part, we will have a Q&A session. And just to let you know, how is this main part structured? So first of all, I will start how visualization can help, or maybe why visualization could help. Then I will talk a little bit about how medical coding quality can be visualized. So we just want to give some hints how we did it, and then at the end, I'm presenting the results of our internal experiment. So as already mentioned by Carol Ann, we all know that data quality is crucial for patient care, patient safety, and for us here in the pharmaceutical industry, for the research and development of our medicinal products, right? And in this context, of course, the process of medical coding is important to ensure that accurate and consistent representation of health data is available, as mentioned by Carol Ann, for reliable data analysis, and then for informed decision making, and of course to ensure that the medicinal products are safe and effective. So we also know that errors in medical coding have serious consequences, such as missing or incorrect diagnosis, ineffective treatments. So if we look outside of the pharmaceutical industry, it can lead to billing inaccuracies, and then of course it would also impair our research quality. And nevertheless, we also know that consistency in medical coding, as shown a little bit by Carol Ann, is challenging, and because we have an increasing volume of data, we have complex terminologies like MedDRA, WHO drug, ICD or SNOMED. It's not just related to MedDRA. And then we also have seen, right, that also we have maybe a lack of quality in the source data. And nevertheless, there is also, especially in the pharma industry, cost pressure and time pressure. And we also see the shortage of skilled labor. So as mentioned, or as presented by Carol Ann, we also use this data, and just to let you know, you can see here in this little picture, in the middle of the slide, how we receive the data reviewed by the MSSO. So as mentioned by Carol Ann, the typical standard approach, right, is utilizing Excel. And we can see here, or in this spreadsheet, you can see, no, you cannot really see because it's so small, just to let you know how we received it. So we received on the one hand the MedDRA hierarchy in column A and D, and then we received some comments and proposals how it should be coded. And we thought, okay, maybe we can also find more or less the same errors as detected by Carol Ann and her colleagues, by using visualization. So this was the basis or the basic idea of our experiment. So next slide. So as already mentioned, so can you, you can see it. So the standard approach, right, is a line-by-line review, checking the reported medication error and the assigned MedDRA code with an Excel to ensure or to check whether the data were coded correct and accurately, consistent and completely. So these are typical, the quality attributes of codes. And we also know that utilizing Excel is somehow time consuming. Yeah, so as mentioned by Carol N, they have reviewed 1,500 records, but we also know, right, if we look at data in our clinical trials, especially phase 3 trials, we may have larger spreadsheets, and reviewing data that way creates a cognitive load. And we also know that ensuring consistency within one large spreadsheet is challenging, also if only with one person doing the review. And there are also limitations in the use of Excel when you try to filter and in large Excel spreadsheets. And it is not easy if you have a lot of terms in a spreadsheet to identify inaccurate or inconsistent data. So sorry, it's a bit animated. I forgot to remove it, so let me do it. So, okay. So you have seen with the presented example and issues, yeah, that or with the presented issues of challenges that beneficial quality control or assurance instruments become more and more essential. And maybe from your own work or when looking outside of the pharmaceutical industry into other healthcare sectors, such as hospitals, you can see an increase in the utilization of visualization tools and techniques, because it has been proven that complex data can be much better and faster understood by healthcare professionals through the visual insights provided by these tools and techniques. And you may also know, right, that the ICH encourages the use of visualizations, especially for large data sets. It's mainly on data analysis and MedDRA output, but nevertheless, right, they state, yeah, in paragraph 3.1.1, so I have this on my slide here. Graphical displays can be useful, especially with large data sets, as mentioned. And then they also give an idea, yeah, what kind of graphical displays, or I call it visualization techniques, can be helpful. They say histograms, bar charts, pie charts, and things like this. So if you now think about that the quality attributes or that you want to detect, yeah, inaccurate or inconsistent coded data, and also you may want to check is the data completely coded. So also Carol M and her team or her colleagues became aware of split issues. Yeah, we thought about how can we visualize this, excuse me, and we more or less translated what is in the ICH guidelines, also in the general MedDRA coding guidelines, and also according to our own company guidelines, coding guidelines, a visualization strategy. So this is what you can see on the right-hand side of the slide, and we started to group this a bit, where we say, okay, first of all, yeah, when you start to review data, you would like to start with a general overview, and then maybe a bar chart might be helpful, where you can see just the number of verbatims per SOC. Then you may then continue with a kind of stacked bar chart, yeah, which is showing the number of verbatims, LLTs, and PTs per SOC. Then we thought maybe it makes sense to have a kind of bubble chart, yeah, where you visualize, have you any terms, yeah, in the social circumstance SOC, surgical and medical procedures SOC, or in the investigation SOC, so just to then check or focus on these kind of data. Then we thought, okay, after we have this general overview, you may want to dive a bit deeper in, is there any wrong or vague PT? So on the one hand, you can start with a visualization of a bubble chart where you maybe visualize if you have any PT that includes NOS, and then which verbatim, and then a second one might be a bubble chart or so which is visualizing failure PTs with the verbatims, and so on. And then you can also go deeper into the hierarchy. Then you can also do this kind of exercise for wrong and vague LLTs. And of course, you can also, depending on your product or how you do your quality control, specify your visualizations or your visualization strategy. So for example, or when we code PR, also procedure related terms, so then you expect just 2 SOCs, and you can check, is everything coded to this expected SOCs. So just to explain what we did. Today, or to visualize data, you normally need expert knowledge or advanced programming skills, and information visualization. Because on the one hand, you need to think about, do I want to visualize this as a bar chart? Do I need this as a bubble chart? Would be a scatter plot sufficient, or is a pie chart helpful? However, so this is how it works a lot of years, so to say. But there is also an increasing market, for tools where you don't need this kind of expert knowledge or advanced programming skills. And you may have already experience with ChatGPT. So in this area, also tools have an increasing, let's say, attention, which utilize or which use large language models for the automated generation of visualizations. So in our experiment, we decided to use Power BI on the one hand for, let's say, this for more engineered visualizations, and then we also decided, let's have a look into and explore LiDAR. So both applications, softwares, however you want to call it, are provided by Microsoft. And so there is, both both applications or tools or softwares are available at BEAR, right? So there was no other particular reason why we have decided to utilize it. So this does not mean what we show here, this is your ultimate way to do it. It's just an example. So of course, there are also other powerful visualization tools like Tableau. It's just an example of what you can use. So I want to quickly introduce LiDAR, and then also I give a very, very short introduction of Power BI, and then we start a quick live demo, so to say. So maybe not many people have so far heard something from LiDAR. So this is an application developed by Microsoft Research. So they also have a research department, and it was shared or published only last year in 2023. And if you want to have a reference to this tool, the very last slide of the shared slide deck includes all references where you can see where we get all the information this is included, or which is included in this presentation, get from. So LiDAR stands for Language Interface for Data Analysis, is an advanced tool which is designed to leverage large language models to automatically generate data visualizations. And so the purpose is to make data more accessible by translating complex data sets into comprehensible visual formats, yeah, and it should help to reduce, yeah, the cognitive load, yeah, of user experiences, especially when you work with raw tabular data. So as you can see on the slide, this application consists of 4 components, so to say. It's just to give you an idea how this is working before we look into it in a live demo. So it is starting with a kind of summarizer. So this module handles the data ingestion, so the upload of the data. And the first step is to create a natural language presentation of the data set, and then, yeah, a large language model enriches the information by adding semantic context. So for instance, numeric fields may be described as dates of stock prices, yeah, and things like this. So yeah, the enriched data, so to say, is then the foundation of the subsequent visualization steps. Then the next one is the so-called goal explorer. Yeah, and here, yeah, the, let's say, interesting part of this kind of application starts. Yeah, so it is creating, or it is making hypotheses how you can visualize the data set you have uploaded. Yeah, and this helps you somehow, yeah, or it is a kind of guide, yeah, for the visualization process. Yeah, so when you have uploaded a data set, I will show you this in a minute. It, yeah, just is making, yeah, some thoughts on how you can visualize your data. Then you have the Swiss generator, yeah, and this module of the application is then really the one which is automatically creating visualizations, yeah, based on the hypothesis you may have chosen. Here, for those who are experienced with visualizations, LIDA is supporting different libraries, yeah, visualization libraries like Matplotlib, Seaborn, or Altair. I have to say, yeah, so the best or nice looking visualizations you get with Seaborn. So I don't like, or I have the impression that if you use Matplotlib or Altair, the visualizations look a little bit outdated, so to say. Yeah. And then it also has an infographer. This is an optional feature, yeah, that transforms the traditional visualizations into more aesthetically infographics, so to say. It is, or it was not available when we have used this, yeah, this optional feature, so I cannot show you, yeah, in the live demo how this is working. So here, next slide. So Power BI, maybe most of you are already familiar with Power BI. Yeah, so Power BI, of course, is a powerful business analytics tool. It's also developed by Microsoft, and it allows users to connect, analyze, and visualize data from various sources. And here, yeah, you may know, in an interactive and dynamic way. We will show you this in some minutes. And in the context of data reviews, yeah, for coded medical data, Power BI could be a valuable asset, yeah, in identifying and addressing coding quality issues, yeah, because one of the major strengths, so to say, of Power BI lies in its ability, yeah, of handling large data sets, yeah, like Excel. So also we cannot demonstrate this, but you have also this kind of Power BI mobile, like you can see on the slides, yeah. So whatever you see on your screen, yeah, on your laptop or whatever, it's also possible to transfer this to a mobile phone, yeah. And this is especially in the hospital environment or wherever helpful, yeah, if a physician or whatever wants to review something and maybe has at this moment no access to a laptop or whatever. Yeah. So, and Power BI, yeah, has specific visualization capabilities for various types, yeah, of visualization techniques, such like, yeah, bar charts, scatter plots, heat maps, flow diagrams, so a lot of things. Yeah, and I think with this, I will stop the presentation here and will try to show you some examples by using LIDA. Maybe some limitations up front. Yeah, so of course, LIDA is using large language models, and you may have also experiences with ChatGPT or other GPT utilities. Yeah, you always need to control what you are doing. Yeah, so it may happen. I had this once, yeah, that maybe such a tool is offering you or is creating, or it's creating a hypothesis with a kind of gender pyramid, yeah, because it's just translating, yeah, in your medical text. So in the as reported term, you may have something like 32-year-old female was vaccinated with, or something like that, right? And you may have other things, 5 years old boy or something like that. And yeah, we know, right, these large language model based software or this kind of tools can have the tendency to make wrong assumptions. Yeah, and then of course, so even, yeah, it's trying to create a gender pyramid, although there is no specific gender information in your data, just in the as reported term, and you have not an extra column where you can see the gender for each record. So this is an observation I had, but just one time you can also see, or it's also a hint that these kind of tools can learn, so to say. So let me stop the presentation and let me go to I know it's very tiny, but I hope so that you can nevertheless that you can see that you can see it. So if not, yeah, as mentioned, so the so a demo version is publicly available. Of course, it will take a bit time, yeah, to install it on your end, but I hope I can show you here some advantages. So let's start here. So I'm here, right? So this is so you see here my Genesis LIDA. So this is definitely something we have at Bayer available. Nevertheless, yeah, also if you go to the public version, it looks like this. Yeah. So please then click on the demo version or on the demo tab, so to say. And before you start, yeah, you have, for example, here the possibility, yeah, to check, yeah, with which library you want to work, as I have mentioned, right? So, for example, you can have Altea, Matplotlib, ggplot, or Seaborn. And it's not good visible here, but you can also choose, yeah, a lot of other settings, for example, with which large language model you want to work. So currently we have just one model available. It's GPT, you can see it a bit here, GPT 3.5 Turbo. so just in case you want to also do it. So as mentioned, so I like Seaborn more than the others one. If you click here, yeah, you see here, click or drag your file on this area. They have already uploaded here, yeah, so files you can explore with or you can test with, and you can also see here, yeah, so LIDA is supporting 2 formats, CSV and JSON. So our experience was that it is better working with JSON. Yeah, so but maybe you made other experiences. Maybe it depends a bit from your tabular data. So you can see here that I have already uploaded, yeah, the data. So the UMC Wikibase data, which was reviewed by Carol N. So it's just the name of the file I have uploaded. And just to let you know, yeah, so the data were coded in MedDRA 24.0, yeah, so you can see this here, but it's not. You can, or we will not check this, so it's just for you to, or just as an information. So when you then scroll down a bit, yeah, so and you have upload your file. As already introduced, it started with this data summary, yeah, and you can see what kind of columns your uploaded data set contains. Yeah, so you can see it contains a column that is called original investigator term. And if you click here on few examples, yeah, you can see how an investigator term looks like. Yeah, then because we have used our format, yeah, how we usually do our review, and that is the reason why you see here maybe columns you are not familiar with, right? Because it's just our format how we do it. So we have a clarified investigator term, yeah. So this is here, of course, just a one-to-one copy. You would not see anything else. And then, for example, in the modified investigator term, so let me scroll down a bit. You can already see, yeah, some selected LLTs. Even, yeah, we have the low-level term, but yeah, we also know, right, when you do medical coding, you can apply a synonym, yeah, and that's the reason why we have the modified investigator term, yeah, because it can be different to the low-level term if you have used the synonym. What you can also see here, yeah, so you can also see the number of unique values, yeah. Usually, yeah, it should be the same. We have here a slight difference of two terms. I have not checked this because it's just a demo, right? So I have not looked into detail why we have here 2 differences. It should be always the same. You can also then see here, right, so that in this data set you have 207 different unique low-level terms. You can directly see, okay, yeah, I have 99 different PTs, and you can click here, as mentioned, you can click on examples, right, and it's just showing you 3, but nevertheless, yeah, you get already an idea. And then the same, yeah, you can see then the high-level term category, yeah. So in this data set, the data were coded in a way that you would have 11 different HLTs, and as presented by Carol N, it was just 1 extracted HLGT and 1 SOC. Yeah. Then the next part is this goal exploration, what I explained, right? So here, LIDA is, as mentioned, right, is really analyzing the data set you have uploaded and tries to offer a hypothesis what could be of interest, yeah, to visualize. So it started, what are the most common types of medication errors reported, and when you click on one of the goals, yeah, the visualization is directly reported here. So this is something I have tried. I will show you a bit later, but I will, so you see here what I have here. If I now click, yeah, on this goal, it is creating automatically, yeah, without doing something. What is this saying here, right? So it's saying it will create a bar chart of preferred term, yeah. The visualization will provide insight into the frequency of different types of medication errors, yeah. So you see here, so it, as mentioned, it has been created automatically. I have to say in this demo version, I'm very sorry, we cannot, so you can make this a bit bigger if you click on it, but it's not, let's say, in a way that you have a full size of the picture, unfortunately not. And you cannot, or I have not found it out, how you can maybe remove this Python code window. So if you think, I'm not 100% sure, what does it mean? You have here the opportunity to explain or to get an explanation of this visualization. Yeah, so and then you click explain the chart. Yeah, and then, yeah, you can, it is then hopefully, yeah, it is an understandable explanation. Let's check here, the section applies to data. Of course, you could then say, this is not so helpful for me because you cannot see anything, right? So because you see here nice numbers, but here it is, yeah, not visible. So you can then review, is there another goal which might be of any interest, right? So we could of course, yeah, if you want to visualize data quality, a word cloud might not be helpful, but just to show you that this is possible. Yeah, and that was the reason why we have started to create it, the visualization strategy, because to really have an idea, yeah, what should be created or what is helpful. Yeah, here it's, and then you see, yeah, it is out of a sudden creating you a word cloud by the data you have ingested. So so on the one hand, what you also can do, if you say, I don't like these kind of goals or hypotheses, you can also create your own one. Yeah, so you could say here, yeah. So if you go, let us go back to the first goal, and was a lot of data. Okay. So we said, right, we cannot see much, and then we could also say here, yeah, you see here this little generate. You could also say, okay, create a bar chart. So this is the beauty of these kind of things. Create a bar chart. Sorry, of the 25 top preferred jobs. And then you say generate. Yeah, unfortunately, it takes a little time, but you can see here now, right? So you just type in your own hypothesis and you click on generate, and you don't need any Python programming skills, right? It is, so of course I can also click here. So then it starts that you see here truncated text, but you have the possibility, right, by using the refine bar to type in prompts, so to say. Yeah. So for example, you could for example, you say, it's nice that I get it visualized that way. You can then say add, for example, add concrete numbers to bars. Takes a while. So I hope you can see it. So and you can see now, right? So first of all, you maybe missed the concrete numbers. And now, yeah, you can see, okay, these are my 25 top PTs. So I guess we had, if I remember correctly, 99 different, right? And then you could say, okay, what is the most frequent PT? Without filtering or something like that. And you can see here, okay, obviously 482 as reported terms, so to say, have been coded to product administration. So as mentioned, you can try to refine it in a way that you can see this as well, but this would take too much time. So it was just to give you an idea, yeah, how this is looking. So it's definitely possible to see everything. Yeah, so and as mentioned, so now you are able, yeah, with these kind of tools, on the one hand, to get an understanding a bit faster of your data. You can start grouping your data by using this, and then you can relatively fast see, okay, you have definitely one PT here where a lot of data, or sorry, a lot of records has been coded to, right? And then, yeah, you then see here, yeah, the other ones are, yeah, coded much less, right, than this PT. Okay, and I think for the short live demo, this is enough, and I would hand it now over to Adrian to walk you through our Power BI visualizations, and then I take it over, or then I take it back again to walk you a little bit through our results. So.
Okay, can you see my? Okay.
Yes, we can see your screen. Mm-hmm.
Okay, wonderful. And also just not the whole screen, just the web browser, right?
We can see your whole screen, yeah. Mm-hmm.
The whole.
So the just the UMC or just the just the PowerPoint. Yeah, but everything for me looks fine.
Okay, yeah. So this in this case, what we did, as we saw that NIDA might not be the tool to choose for us, we took Power BI as an business intelligence tool to try also to visualize this vigilance data, or in general coding data, in a way that inconsistencies or errors are being found faster than using an Excel sheet with thousands of lines. And we tried out different kinds of approaches to visualize them. Power BI has a bunch of different visualization built in you can use, yeah. If you have your data once in the tool. And this was our idea how we could support the coders with this review. So first we have here this kind of heat map, yeah, where we show top 20 most used PTs. And the good thing about Power BI is, for example, if I'm interested in a specific PT, I can just click on it, yeah, and this would then filter everything else on this page. You see, so I clicked here on the PT wrong product administered, yeah. And what I have here on the bottom right is a detail table where I can really see these 26 records, yeah, which were coded to this PT. So I can see the investigator term, clarified investigator term. This might be via wording, yeah. So, but it's in the end there's reported term. I can see the LLT it was coded to the PT and the whole MedDRA hierarchy. Yes, so can click through all of this. This is now very small, so if I want to see it a little bit bigger, each visualization within Power BI can be switched on to focus mode. So there we go. Now I have just this table filtered with this one PT, and I can have a look if there's some funny verbatim or as reported term which does not fit. Yeah, so I can go from this direction from PT to as reported term. Yeah. Then we did the same with the LLTs. So we have the same kind of heat map with the most used LLTs. Yeah, out of this data set, which contains, you see here on the top, 1,455 records. So also here we can even go and focus on this graphics to see a little bit more. Yeah, and then say, okay, this one, for example, is interesting for me. I click on it. Yeah, I go back to the report, and then in this table, I can see just everything regarding this LLT, yeah, which was coded to this LLT, and have a better look. Yeah, so we have a kind of grouping with this visualization. You don't have this 1,500 records right in a list and don't know how to start. So if you get lost in your filters, like currently you can just reset all the filters of the whole page. Yeah. So now I have everything again. What we have here on the top is, as I said, is the number of records in this data set we use, yeah, which is the vigilance stuff, which Mark used for his studies. We would have here a view, which in this case is not so useful, but we would see the distribution within SOC, yeah. So we have here the verbatims, yeah, and these verbatims were coded to 207 LLTs, yeah, from 99 PTs, yeah. Unfortunately, we only have this SOC. Normally, if your data would be spread over more than one SOC, you could see that this here, yeah, which could also be a hint that something might be coded wrongly or inconsistent. Yeah, in this case, we only have this line, unfortunately. What we then also tried is the bubble chart, which is a bit similar like this heat map stuff. Yeah, so we took here a bubble chart showing the top 20 PTs by its used LLTs. So we have here this LLT, yeah. We have the top 20 PTs and see here this one, for example, this from this PT incorrect dose administered, 38 LLTs were used, yeah, in this data set. And also with this bubble chart, we have exactly the same filter capability. So we can filter here, go back to the report, go to our table. Yeah, table is also configurable as we want. So we can add several columns very quickly here. Can sort, yeah. So we would have now this data. Then we have the second page here, because this was a little bit big. What we did here and on this page, for example, we ran our data against an SMQ. In this case, the medication error SMQ. So out of our data set, which is mostly medication error, this amount of PTs was coded or is part or is a PT which is contained in this SMQ. And also here we created a heat map. Yeah. So if I click on one of these items, I can see here on the bottom the LLT, yeah, and then the verbatim it was coded to. Yeah, so it's 90 records left. Here we have another pie chart, yeah, with LLTs containing the word vaccine, yeah. So even on a pie chart, I can use filters. So if I click on a part of the pie, yeah, you can now see all LLTs which contain vaccine, which were used in my data set, and the corresponding verbatims, and look from this perspective on my data. So it's possible to look from different perspectives on the same amount of data. And this is the way how we try to say ease this review process. So this would be already everything from my side. Thank you. I will hand it back.
Thank you.
So I think we'll be going back to Marit now.
Yes.
Fantastic.
Yeah. So. So you have now seen, right, our 2 live demos, and as mentioned, yeah, so we present now the results of our internal experiment. So we did, yeah, on a, let's say, scientific perspective or academic perspective, a within-subject quasi-experiment. So so our experts, yeah, 6 have reviewed. Sorry, sometimes I forgot to stop this kind of visualization, to stop this kind of animations. So as mentioned, yeah, we used this 1,500 COVID-19 vaccine records. We brought them in our visualization experiment. We used the shown visualization techniques, yeah, so LIDAR and Power BI. And in this experiment, we included 6 diverse experts. So it was an experiment for medical coders as well as for our physicians, yeah, and we did this in some steps. So first of all, yeah, so we have taken the data provided by Carol N and transformed them a bit, yeah, as mentioned in our format, how we do, yeah, the Excel review of clinical trial data. And then this Excel report was provided to our review in the classical way, yeah, the line-by-line review utilizing Excel. Then we reviewed the data by utilizing LIDAR, and then, as shown by Adrian, we did the same review again by utilizing a Power BI. And at the end, yeah, to really get feedback from the participants, or myself, yeah, so I did an interview with my colleagues to really get the feedback, yeah, so not just to explore, but also to get feedback how they like the different visualizations. So this means we discussed, right, the benefits of what we did so far, yeah. So we went through a kind of semi-structured questionnaire, and we have asked them, or I have asked them, to give me feedback on categories such as clarity of visualization, understandability of visualization, was the data traceable, was the visualized data correct, and things like this. Okay, and here the results of our experiment. So LIDAR, the understandability, traceability, and correctness, also these 3 categories were assessed as good, yeah. However, half of the participants found the LIDAR visualizations less clear, and you may have seen this already, right? So sometimes you cannot see, you have this little window, you need to squeeze things in, or you need to somehow think, okay, what was mentioned here, yeah. And by Power BI, yeah, you can see already more or less the pattern, yeah. So Power BI visualization seemed to be a bit more beneficial. Yeah, so almost all categories get a, so to say, yeah, very good or good, yeah, in these assessments when we talked about clarity, understandability, ease of orientation, and things like this. So overall, yeah, so after the interview, nevertheless, yeah, 4 out of 6 participants stated that a leader is somehow helpful, but it was limited at the end. What you may have also seen, yeah, it definitely helps you when you start with your review, yeah, to give you an idea of what kind of data do you have, how many NLTs, PTs, and things like this. So yeah, how is your data structured? But there were also 2 participants. One said it was not helpful for me, and another one said for me it was less helpful. And so with Power BI, yeah, it was a bit different, right? So all of the participants either assessed it as helpful or very helpful. Of course, we also need to be fair, right? Compare LIDA as a kind of large language model based application, compare with a complete, right? Business intelligence software is not fair, but it was just an experiment to see, right, how these applications where you would not need programming skills or advanced knowledge about visualizations, whether this is somehow helpful. Okay. And what we also did, we also checked, yeah, were we able or how much findings or, yeah, so as you may have remember, yeah, so the review of the MSSO colleagues, they categorized their findings in different error types, yeah, as mentioned by Carol Ann. So error type one was unavailable, when maybe it was reported in a different language, so not in English, or it was truncated or something like that, right? And then, as mentioned, incorrect PT or incorrect LLT, and so the first, yeah, part of the bar is the findings or the number of findings by the MSSO, and then what all the other or what our participants then found when they did the review by using Excel. Yeah, and just to let you know, in this experiment, right, we limit the time for this review to 8 hours. Yeah, and that is the reason why the numbers, yeah, of findings, yeah, collected by our experiment participants is not exactly matching what was found by the MSSO colleagues. So I have the same or a similar chart for the assessment when we use Power BI. Yeah, so the colleagues were definitely able to identify, as mentioned, data which was unavailable, or which was incorrect coded from a PT perspective, or incorrect coded from an LLT perspective. I also have to say, so I cannot present this for LIDA because we had also here on our end some technical hurdles. It was not available to all the participants, or would have brought installation efforts with, where we thought, or where myself thought, maybe this is too much. So I have presented the visualization strategy, and this was the basis to create all the listed visualizations there with the help of LIDA. So this was more or less an experiment for myself, yeah, because I don't have Python programming language and also not much visualization knowledge. Yeah, so I was able, yeah, to create the visualizations defined in our visualization strategy with the help of LIDA, but nevertheless, these visualizations were provided to our participants in a kind of static report, and this, of course, maybe led to a bit of finding LIDA not so beneficial or not so nice as Power BI, because it was simply not possible to interact with the data or with the visualizations. So overall, we said, or we found that visualizations fully or semi-automatically generated by large language model-based software like LIDA, seem to be helpful, but we also assessed, or the colleagues also assessed, that it is less helpful than Excel. But when we did the assessment for Power BI, it seems that visualizations generated by business intelligence tools like Power BI, that they are helpful in detecting coding inconsistencies, and that they may have the potential to replace the Excel-based process. Our participants, so our colleagues, so the medical coding team at Bayer, so they gave already some feedback how the visualizations of what has been presented by Adrian can be improved, and what would be some requisites to fully replace maybe the Excel-based process. So with this, I like to thank my colleagues from the medical coding team at Bayer for their support and willingness to explore these new ways of working and new tools. And with this, I hand it over to Uta for the last minutes before we start with the question and answer session. And Uta will now share her experiences as she was one of the participants in this experiment. Thank you very much.
Thank you, Marit. Yes, as you have mentioned, I would like to briefly share my experience and my impressions from taking part in the experiment. And before I go into the details for the experiment, I would like to mention, can you hear me?
Yes.
We can indeed, Uta.
Oh, sorry, I was confused now.
Oh, sorry, it was my fault, Uta.
Okay.
Because I stopped accidentally the presentation, I'm sorry.
Oh, okay, no problem. So first I would like to mention that at Bayer, as I was involved in medical review of coded clinical trial data for many years, and the format to collect and present the data to be reviewed we use here is an Excel spreadsheet with filter options to separate auto-coded from AI-coded data accepted by our medical coders. This manually accepted data set is then reviewed for coding correctness and consistency, and it is a full review for adverse events reported in phase 3 clinical trials. Such data review in an Excel file might become very exhausting and time-consuming for the coding correctness alone when the number of records in a report exceeds 500 to 1,000 records, but it is particularly challenging if the report includes more than maybe 10,000 or even up to 30,000 entries, sometimes like for large-scale clinical studies. However, and most importantly, this sound verification of coding consistency is hardly possible with this amount of data, since it is highly difficult to keep an overview of similarly reported events. As Marit has presented earlier, the first quality control report we have reviewed during the experiment was the Excel file alone, and I remember that it was challenging to complete the data review in the given time frame. The LIDA report was a very good introduction to data visualizations. However, receiving them in a report using a Microsoft Word format was less suitable for the content review because it contains so many pages to which one has to go back over and over again. And this repeatedly has caused losing focus on the information intended to get. The part of the experiment I liked most was the second data review in an Excel report, which was supported by Power BI data visualization. Its speed of the individual data views was surprisingly fast, and the dashboard was very clear. The visualization has helped me a lot to perform the data review for coding correctness much easier and faster than doing it in Excel alone. Since as a reviewer, you simply see where to dive in deeper to identify incorrectly coded records. However, the visualization was particularly helpful in checking the consistency of coded data, and its positive impact will become greater the more data sets are included in the quality control report. Finally, I would like to mention that the data review supported by visualizations was not only less exhausting for me as a reviewer, but it was also more fun instead. With this, the floor is open for questions and comments from the audience. Thank you very much.
Okay, well, thank you, Uda. There are several questions. The first question is, I think, more of a point of clarification. The question is, you mentioned looking for PTs that contain NOS or not otherwise specified. Are you using a custom MedDRA dictionary because the standard MedDRA dictionary does not have any NOS terms at the PT level?
This is going to Marit. Am
Yes.
I correct?
Yes.
Yes. So we just used how we receive the data, and as mentioned, we transformed it into our Excel format, and then we can see it. But nevertheless, when you try to do this kind of visualization, you also you have not seen something like this. It was not visible. But in case you have synonyms or something like that, then you would be able to see it. Mm-hmm.
Okay. So the next.
Hi, this is Martin. Yeah. I've heard this and seen this myself, and had been puzzled, but actually Mara did not mean that there are PTs that contain the NOS, because that doesn't exist. It's on the LLT level only. But I think she was looking as PT where the data set has LLTs under it that has been used with an NOS. So that was actually the precise formulation of what she analyzed.
Thank you.
So everything all right. The technology was correctly used, and MedDRA was correctly used.
Okay. Thank you, Martin, for that clarification.
Yeah, thank you.
So let me move on to the next question. So there's this is actually a bit of a several questions here. So I'll read it and you can respond as best you can. So the focus of the question is regarding the implementation, a straightforward implementation of a system for a company that's just starting out and needs to be simple. How would you recommend setting up a basic data analysis process? For instance, could exporting MedDRA coded data to an Excel sheet and then cross-referencing it with codes recorded for a specific drug in the database be an effective starting point? And do you think a search C, search V approach in Excel would work for flagging potential safety signals, or would you suggest a different method for more reliable results? So quite a bit in that question there.
So shall I start? And then maybe also Uta, if you like, you can maybe also add something from your experience as a reviewer. So here it was just a start, right? So usually we would not, so so our clinical trial review process, would not look into this kind of data. It's really to check, is all data correctly coded. So as mentioned, it was a kind of experiment, and of course you would definitely need to transform the review process in a way that it is efficient, using this kind of visualizations. Nevertheless, so we cannot show you, let's say, our product data. So the visualization strategy we have presented is something we have available on our end with clinical trial data from our clinical trials. And as mentioned by Uta, so you can definitely see things faster, right? So I hope this answers a bit the question, but maybe Uta, you want to add also something, or maybe Karen.
Yeah, Marit, thank you. I fully agree. So the Excel file to be reviewed is a valid approach, but the visualization will definitely help in doing the review more fast or more easy, and will of course support the consistency checks, because this was the most important part for me. This is not easy to keep an overview if you have thousands of records in your report, and the consistency check is much more easier with the visualization, as I have experienced it in the experiment. So, but we will continue to use Excel files in future, I would assume.
Yeah. So, and maybe what we could also add, so of course there are also limitations, right? If you have a phase 1 study and just 20 records, it makes no sense, to use visualizations. So as mentioned by, or hopefully, it was a bit clear. So we would use visualizations, even maybe I said it, replace the Excel based approach. It's something good, right? And it needs to be seen in the perspective or context of large data sets.
Yes, I fully agree.
Okay, any more comments on that question before we move on? Okay, well, the next question is a pretty straightforward one. Is it possible to export these reports in Microsoft Excel?
Yes.
Okay.
You can export parts. So what you saw in the Power BI, for example, this table, you can export just the content of this table into an Excel sheet. That's possible. And what we also do, we use another Microsoft tool, which is Power Automate, which is part of this Office 365 suite, to automatically do this. So we query the Power BI report and create an Excel sheet more or less automatically every week for review. In other examples. So this is also possible.
Okay, thank you. So the next question is focused on Power BI. The question is, Power BI was rated as correct by 67% of the expert pool. Was the lack of correctness a feature of Power BI or a feature of the data engineering behind Power BI?
Maybe you can repeat the question, so sorry.
Okay. I think this is something about Power BI itself versus how you're using Power BI. So I'm not sure if you'll be able to answer it, but the question again is, Power BI was rated as correct by 67% of the expert pool. Was the lack of correctness a feature of Power BI or a feature of the data engineering behind Power BI?
No, so what, so it was maybe not good visible in this kind of presentation. So what I presented in this, let's say, charts, it was just the highest rating, right? So it was taken, or this kind of information was taken from an interview, and if 67% maybe said correct or very correct, maybe, yeah, so I have maybe not received the answer in the interview by 2 colleagues because we just not went over or it was packed in something else. So it is, yeah, so we need to be also a bit careful if we say it was not correct, or if the impression gets here, or if you get the impression it was not correct, it was just really, yeah, or how I displayed the data here, yeah, that I just checked, yeah, what kind of, so when you do these kind of interviews, yeah, you have a rating scale where you are a scale, right, where you say absolutely correct, and then, right, so it also, yeah, depends on your interviewers or on your participants with which answer they feel most comfortable, right? So this is how you have to interpret this, what I have shown here. I hope it makes sense and you have understood what I mean.
Okay, thank you. Any additional comments from the group on that question before we move on?
Yeah, hi, this is Martin again. I understand the question also in the way, is it really reliable to allow a tool like Power BI to do this type of analysis alone? And naturally, these tools are all as intelligent as they have been programmed. So if you have chosen a visualization that allows a reviewer to identify a certain strange type of coding, that is one set, and this needs to be programmed somehow. And you cannot program something you do not exactly that you don't know. So it will always be or it will never be able to find all the errors because you don't know all the errors that can be made. And you don't know how to program it to then actually pop up in a visualization. But it's a start, and it may make things much easier, especially it may, the well-known errors will pop up right away and maybe analyzable very quickly, and so you can start a respective corrective action for your team, whatever you can do, so to get at the root of the issue.
Yeah, I think it also depends a bit on your, let's say, review strategy, right? Of course, if you have to review 100% of your data, you need to somehow ensure that you have access to 100% of your data, right?
So, also to add, Power BI is just a reporting tool. It does not contain any AI. Yeah, so it's just showing the data in the form developed. Yeah, so to show the data. It will not tell you if something is right or wrong. It can give you a hint, a visual hint, where to look. Yeah, but in the end, the coder or the expert has to decide if this is coded correctly.
You only see what you're ready to see.
Yeah, but
and that's what Power BI will help you. You need to make it show you things, but you need to know before.
Yeah, it's known by reporting. So the reported information, including all further details, coding, and so on, are analyzed with Power BI and are only represented in a different way, in a visualization. So it's not referring to any correctness rates or whatever. No? It's only a different way of presentation.
Yeah. So it can. So it's not that. So the question regarding correctness was: had the reviewer the feeling that the visualizations presented has correctly presented the data? So this was the question behind, not whether the data was correctly coded. Yeah.
Exactly. Yeah.
Okay. Any other thoughts before we move on? We have 2 more questions left. So the next question is, how do you compile the results of the 6 study experts presented on the bar charts? Since the reviewed sample size seems to have been different, how was the consensus on the results reached?
Yeah, on the one hand, right, from an academic perspective, we did a case study. So this means already that generalizability of these results is maybe not possible or is very limited. As mentioned, so on the one hand, our review time was limited, and on the other hand, what we not include was those records where the MSSO had assigned a split issue. Yeah, so because it was just not, or it was not, let's say, for us. So I just excluded them because I thought this would create, yeah, too much thing, or it was just complex. Let's say the task. Yeah, and therefore we have reviewed a little bit less of the data. So, and this is what I also tried to mention. Yeah, so we did a review, and I think we were able to detect similar issues and a similar number of issues, but we also know, and maybe Uta, you want to comment on this? Yeah, each reviewer may have a different opinion.
Yeah. Asking for doctors, you will get 5 answers, or how is the thing?
Yeah. In the interpretation of the results. Yeah.
Yeah, that's right. And maybe we have had also different level of experience and speed and so on. This might also be
data, right?
a reason.
Yeah.
Yes.
Yeah. Yeah. So this was also feedback, yeah, from the participants that this kind of review, I hope I mentioned this, yeah, so medication error for vaccines is usually not a common data set we review, yeah. So, and thank you, Uta. Yeah, I think this was also a bit, I don't want to say limiting the results, but yeah, it was definitely feedback by you and other colleagues which participated that the data was a little bit unknown and that it took time to familiarize with the data. Yeah.
Yeah.
Okay. The final question that we have is this really, I think, is just more of a basic coding question that our panelists here can answer. If a coder should always follow the predetermined pathway hierarchy for the primary sock, then why is there a need to show secondary socks?
Did we show secondary socks?
No.
I think this may be a more basic question. I'm interpreting that as.
Okay.
Yeah. So what we just tried at what can be interesting from a medical coding quality assurance perspective, right? So it's not. We do not analyze the data, right? So it's just, yeah, how can we visualize the data to get an idea, yeah, if the data is correct or accurate and consistent coded, right? Because, yeah, we know, right, MedDRA is complex. We have a lot of changes with each MedDRA version update. This was also, yeah, a reason behind where we thought, okay, let's check. Yeah, of course we do a lot of refresh with our data, or our data gets refreshed, but nevertheless, yeah, we know that MedDRA is complex, and we would not. So for this kind of visualization or for this kind of review process, we would not, yeah, visualize or use or display. Also, not in our Excel approach, second socks or secondary paths.
No. Yeah, only the primary paths. Yeah, you're right. This is our usual approach, to only refer to the primary sock, but not to any secondary socks.
Yeah.
But that is actually, hi, this is Martin again. This is an analysis way to look at it. For coding, actually, the primary or secondary SOCs usually have not the highest importance, really. When you're really in the coding situation, you should check the allocation, the system organ class, or the complete path, whether it actually matches the reported verbatim or the reported special situation. So this is where you just make a check. And for the details, whether something is primary or secondary SOC and what the allocation is, that is not really that important. It's for the situations where you have like an acquired condition and you end up with a LLT which matches into the congenital conditions. So there is where you check, and then you change and think, well, isn't there not something else available in MedDRA? So that's kind of where, as a coder, you need to look into the hierarchy, and that is not too often. That's a limited number where you need to check. It's a plausibility check for coding.
Yes. But this is in scope of the study teams, for example, to look at these data from the analysis perspective. No? While only coding the data, we just have the primary SOC in our report and we focus on this. Yeah, but you are completely right, Martin. This is important. Mm.
Okay.
All right,
Well, thanks everybody. Sorry, Brian, I was just going to jump in. So yeah, I think it's some brilliant questions, everybody. Fantastic stuff. Hopefully you can all see my screen quickly because I'm aware that we are over. I just wanted to run through the last few housekeeping bits. So the first one is obviously do contact us if there's anything in particular. I mean, there were some interesting questions there which may well lead on to potential future topics in future webinars. So feel free to contact Martin or the MSSO help group with any suggestions for future webinars. Also, if you have a topic, as we did with Marit, and by the way, I'd like to say fantastic presentations, everybody. They're really, really interesting. We've got some good ideas I'm going to steal from those, so thank you very much. So yeah, also, if you'd like to present future topics, please let us know. The final piece then is a call out to complete the survey, which I believe, Brian, you had posted onto the site, onto the questions. And it's also just to let everybody know that we have our next EU MedDRA user group face-to-face meeting in April next year in Warsaw. We don't have the exact dates yet, but please do keep your eyes on the MSSO site for updates on that. And finally, just a big thank you for everybody who made the time to attend, and thank you for the presenters for a very, very, very interesting topic.
Thank you so much also for the opportunity to present this. Thank you.
Yeah, thanks a lot.
Thanks. Bye, everybody.
Bye.
Bye.